Okay, part 2 of the lecture for chapter 39. I did find a better image here. We're going to look at some ways that doctors can determine if you're having some kinds of problems with your lungs. And there's a couple of terms here. Without getting too technical, we'll look at a few things. See if I get my pen. Where's my pen? Pen. Here we go. Okay, so there's this term FEV1. It stands for forced expiration volume, like exhaling. How much, when you breathe out, how much volume of air comes out in the first second. That's what the 1 is. And that number is considered important. So you have somebody breathe into a spirometer, which is like a little tool that you put in their mouth, and see how much they can breathe out. A normal person can breathe out quite a bit in that first second. And the curve here, it's very steep, and then it starts to level off. You have an inflection here, and it kind of levels off. And there's a certain point where they've expired everything that's in their lungs, and that's called FVC, the total amount of air that they can get out. So FVC is the total volume, forced vital capacity. So it's the total amount that you breathe out. FEV is how much comes out in that first second. So both of those numbers are considered important measures. If you have something called an obstructive disease, and there's several things that classify as obstructive disease, we'll talk a little bit about that. You can see the whole curve is more linear, but you don't get near as much out in that first second. However, as you continue to force air out, you eventually get a reasonable amount of volume out. If you have something called a restrictive disease, you get more out in that first second, but ultimately your total volume is kind of about the same as the obstructive disease individual. So they look at these numbers, and they also track them over time for your own data to see what's happening, to try to combine this with some other symptoms to see what might be going on. But a normal person would have most of their volume comes out in that first second, and they can get to a pretty large volume overall. If you have an obstructive disease, literally an obstruction in the releasing of that air, or just a restrictive disease means there's not an obstruction exactly, but something is restricting the total volume of your lungs. So these numbers really is what I'm kind of more interested in, FEV1 and FVC. All right, so make sure you know a little bit about what those are. Okay, so one that's an obstructive disease is called chronic obstructive pulmonary disease, COPD. Although COPD is not a particular disease, it's more of a class of diseases that are obstructive. So there can be several that fall into this, but I list asthma as something. It's where your body releases too much histamine, which constricts the bronchi, and you're not able to, it obstructs the movement of the air into and out of the lungs. Emphysema is also obstructive, and that's where you have less alveoli because the walls between the little alveoli bubbles break down, and you end up with larger compartments. So really what you're getting is less surface area. The lungs are also less elastic, and so it's harder because you have less surface area. You have to ventilate the lungs more often to try to get the same amount of oxygen into the blood as somebody who doesn't have emphysema. So they have to ventilate more often, which tires out the muscles. So people with emphysema become exhausted because they have to keep contracting and those intercostal and the diaphragm muscles more often just to try to get the normal amount of oxygen in. A lot of emphysema is caused by cigarette smoking. It causes the breakdown of the lung tissue, but there are cases where somebody never smoked and you can get emphysema without smoking, but it's much, much more common with smokers. And then lung cancer. Lung cancer is something that comes sometimes with COPD. It's depends on what source you look at, whether they consider it obstructive or just something that is accompanies an obstructive disease. I'm not too concerned about the classification here, but lung cancer is when a cell spreads, starts dividing, and then spreads to other tissues and starts pushing against the other tissues and growing in these tumors that are filling up all the spaces. And really, it's not the cancer, the cells themselves aren't causing the problem, they're just in the way of your normal organs being able to do their functions. So because you have this growth in your organs, it's pushing and invading and really disrupting normal function of other organs. Also, normal function of the lung itself. But metastasized lung cancer has a very bad prognosis, with only 3% of patients surviving after 5 years. And usually 5 years is that, in cancer biology, it's kind of that critical time point. All right, we're going to talk a little bit about hemoglobin, because hemoglobin is what carries the oxygen in the blood. We are crossing over into a discussion, I guess, of circulation, but you can't talk about respiration without talking about circulation. It just doesn't make sense. So hemoglobin is a protein. It's found inside of red blood cells, and you'll remember those are called erythrocytes. There are 4 polypeptide chains that make up one hemoglobin protein. So that's called the quaternary structure of protein. There are 2 chains called alpha globin and 2 chains called beta globin. In the human genome, there's a gene for alpha globin and a gene for beta globin. And so when those are transcribed and translated several times, you can assemble, the cell will assemble hemoglobin. Now each cell has thousands and thousands of hemoglobin molecules in it. The hemoglobin has a porphyrin, actually 4 porphyrin rings in it, and those are called heme. And the heme is what binds the oxygen. When the hemoglobin contains oxygen, it's called oxyhemoglobin. When it's not, it's called deoxyhemoglobin. So this picture shows within, this is a red blood cell, and they're trying to show you that inside of it there would be thousands of these proteins. So this is the hemoglobin protein. You have thousands and thousands of these inside of any one red blood cell. So I'm going to say times thousands. And each hemoglobin is actually, let me see if I can draw a line around it. Here's 1, here's 2, here's 3, and here's 4 polypeptides. 2 of them are called alpha, 2 of them are called beta. I can't tell in this picture which is alpha and which is beta, but they each have this porphyrin structure called heme. This one's kind of behind. Here's a heme there, there's a heme there, and there's a heme there. So 4 polypeptides, 4 hemes, and each of the heme groups can carry oxygen. The iron that you need for your blood to function properly is actually a cofactor. You remember cofactors, a cofactor of hemoglobin. So it's one of those minerals that you need in your diet, and the iron actually attaches to the heme groups, and that makes it function with carrying the oxygen. Hemoglobin without oxygen, or excuse me, hemoglobin without iron is not able to bind oxygen. Okay, so we talked about when the blood picks up oxygen at the lungs, it usually has a partial pressure of about 100 millimeters of mercury. And typically we're talking about something we call saturation, hemoglobin saturation. I don't know if you've ever been at the doctor. They put that little thing on your finger and it measures your oxygen. So they just put it on the outside of your finger. Somehow it measures the oxygen saturation of your blood. So that's pretty normal, 97, 98, 99. They typically, you know, if you're not 97, if you're less than that, it actually they start to get a little worried about you, and they might put you on an oxygen, you know, mask or something like that. So you need to be around 97 saturation of your hemoglobin. Now, when you are at rest, the blood that comes back to your lungs has about 60. I think in the other picture we said 40, but something along that line. So 40 to 60 in what we would call deoxygenated blood, which is kind of an interesting concept because deoxygenated blood doesn't mean it has zero. So deoxygenated blood, I can't spell, oxygenated blood does not have 0 millimeters of mercury of oxygen. I mean, that's just not possible. But it has something, 60 is what it says here, 40 to 60 would be kind of reasonable. All right. So there is still oxygen in deoxygenated blood. So deoxygenated blood is really kind of a bad term. So what we call deoxygenated, just really we should say low, maybe low oxygenated would be a better term. But they didn't ask me. So there's always some oxygen in the blood. Now, if you get your breathing cut off temporarily, it can't go off for too long, but if your breathing gets cut off, you still have a little bit, and you guys probably know, you have a few minutes where you can still be okay. And in that case, it would even drop even lower. But that's kind of a reserve. Or if you start working out, and it takes a little lag time for your heart to increase the pumping and stuff, there's a little bit of a reserve. So it's very small though, it's just a couple minutes of reserve. It doesn't last, you know, indefinitely, but deoxygenated does not mean 0 oxygen. That's kind of important and yet maybe a little counterintuitive. All right, on this chart you see what's called the oxygen dissociation curve of hemoglobin. So this shows how hemoglobin is attracted to oxygen based on some factors. And the normal attractiveness or affinity of hemoglobin for oxygen is the black line. So under normal body conditions, if there's a lot more CO2 in the blood, if there's a lower pH, then that actually causes hemoglobin to have less affinity for oxygen. If there is a lower CO2 level and a higher pH, then that gives hemoglobin a higher affinity. We're not going to talk about the temperature issue, although that is also a factor. All right, so higher affinity is associated with a higher pH. Part of what your kidneys do is to remove excess hydrogen ions. You'll remember that hydrogen ions, if they're in the blood, that would lower the pH because it increases the hydrogen ion concentration of the blood. That would be a lower pH. So the kidneys remove the hydrogen ions so that it avoids a reduced hemoglobin oxygen affinity shift. You may not know this, but carbon dioxide is not carried on hemoglobin. So CO2, for the most part, is not carried by hemoglobin. That would seem may be logical, but it's not true. Hemoglobin does not carry CO2. What CO2 does in the blood, it gets converted into CO2 combines with water, and it makes something called carbonic acid, H2CO3, which then breaks down into bicarbonate atom, CHCO3 plus ion. So this is bicarbonate, which is a buffer. So CO2 in the blood actually becomes bicarbonate ion, and it's carried like that in the blood. When it goes back to the lung, though, it reverses through this process and is released as gas. But in the blood, it's actually converted into this bicarbonate. So, and it's carried outside of the red blood cells. It's not carried inside the red blood cells. So oxygen is bound to the hemoglobin inside the red blood cells. Most of your CO2 is being carried as a bicarbonate ion outside your red blood cells. So that's may not be the way you thought that it worked, but that is how it works. Now, what if you have some kind of mutation? Well, one of the most well-studied and fairly straightforward blood diseases is called sickle cell anemia. Sickle cell anemia is what we call a hemoglobinopathy. Hemoglobinopathy. So a malfunction of the hemoglobin. There are other hemoglobinopathies. One's called beta thalassemia. There's actually an alpha globinopathy. I forget what it's called. But sickle cell anemia is a beta globin issue. So you remember that each hemoglobin had two alpha proteins and two beta proteins to make one hemoglobin protein, and then each of these had a little heme group that bound to iron and oxygen. So the beta globins are an important part of the hemoglobin. If you have a particular mutation, an unfortunate mutation, in the gene that encodes beta globin, then you will have a malfunction of the beta globin and hemoglobin at large. And what this does is the hemoglobins start to stick together in these big clusters. And instead of just having the individual hemoglobins floating in the red blood cells, but not attached to any other hemoglobin that's inside of that cell, they all clump together. So what you end up with is these clumps of hemoglobin inside of the red blood cells. And when things get large, when things clump together like this, they become insoluble. So you get this actually insoluble mass. Instead of the hemoglobin, which is completely soluble inside of the cell, you get an insoluble solid mass inside the cell, and it stretches the cells into these irregular shapes. Now, some of them, they don't have names. Some, when you have one like this, that looks like the kind of the textbook example of the sickle shape. Sickle is an old-fashioned tool that was used to harvest crops. It was a knife shaped like this, but not all the cells are going to look like that. You're definitely going to have some that look just kind of misshapen like this, and so on. But sickle cell anemia is where the hemoglobin inside the cell is clumping together and making an insoluble mass, and that's causing the shapes of the cells to become irregular. But really, the irregular shaped cells, that wouldn't be a problem, except they cannot flow through the small capillaries. So they get jammed up in the capillaries, and you get these blood vessel blockages. So it's really a very dangerous disease. Okay, so the last topic, carbon monoxide, something you've probably heard of. Carbon monoxide does bind to hemoglobin, which is kind of interesting because carbon dioxide really doesn't. Carbon monoxide is what they're showing as CO. So instead of CO2, we have CO. All right, so that's carbon monoxide. Carbon monoxide actually binds to hemoglobin stronger than oxygen. So here's what happens. If you have carbon monoxide in your house and you can't smell it and you can't tell, what will happen is, even if the amount of carbon monoxide is not that much, like 20%, so if you're in a low range here, then what you will have is, look how much the amount of oxygen is dropping. Remember I said when your saturation of oxygen is less than 97, like 97% is where we want you to be. That's this first one. Look over here, you're at like 77. So you've got a problem right there. And that's not even that much carbon monoxide. You'll start to feel sick actually when your oxygen binding gets, in the low 90s. So what happens sometimes is carbon monoxide fills up your house while you're sleeping, and you actually just die while you're asleep, and you didn't even know that you had carbon monoxide. That's why you want to get a carbon monoxide detector, otherwise you'll never know it. But see, it really quickly brings down your oxygen levels because your hemoglobin actually prefers to bind to carbon monoxide more than it wants to bind to oxygen. So carbon monoxide is dangerous to everybody.